going to tell you about the current status of brain pathology informatics, and also I'll bring in some insights from computational and molecular biology that are being used by biologists and neuroscientists like myself in the basic research community. So in the current practice of clinical neuropathology, doctors work with neurologists, radiologists, and surgeons on cases to identify a diagnosis and inform treatment courses on living patients based on biopsies, such as from skin, muscle, or tumor sections, or they might perform post-mortem analysis of tissue, which is what people typically think of as most of the work of pathologists, although this tends to be actually a small fraction of what many neuropathologists actually work on in their caseload. So where informatics currently plays the largest role in neuropathology is in what is referred to as molecular informatics, or just genetic sequencing of tissue samples to provide causal or mechanistic insight, such as for tumor classification. This can also be used either to guide treatment decisions or to provide specific diagnostic refinement. And so another avenue for informatics is cytopathology, where pathologists study cells in solution on a slide, allowing them to study features of the cells themselves, which is in contrast with tissue sections that provide more information about tissue structure and the cellular microenvironment. So just as an example, on the left, there's an example of a solution or a smear versus a tissue section, both stained with hematoxylin and eosin, and both of which show characteristic features of a pilocytic astrocytoma. And you can appreciate that these two images provide very different types of data for image analysis on the same disease. And it's important to point out too that although samples that might be sectioned from whole tissue, as in this example on the right, that they may not be able to be obtained until after a major surgery or even post-mortem, which of course makes it of limited utility as far as treatment of the patient. So currently, cytopathological analysis often is a bottleneck for neuropathologists, and many institutions employ people referred to as cytotechnologists, and these people help with the workload by flagging samples that are most likely pathogenic for review by clinicians. And so, as many of you probably are thinking, this presents a really clear opportunity for machine learning to be employed. And there are several proprietary technologies that exist and that some hospitals that can pay for them use. There hasn't been a widespread adoption of this quite yet, especially in neuropathology specifically. And so one of the barriers to this in neuropathology happens actually pretty far upstream in the process, and part of it's due to the nature of the data. So as many of you are aware, machine learning has been widely applied to studies of MRI data and in clinical data. And although there are challenges associated with maybe the size and formatting and pre-processing of this data, importantly, it's already been digitized. In contrast, pathologists work with histological sections. So the tissue is first sliced, or cells will be put in solution and then stained and placed on glass slides. And then they must be viewed under a microscope. So an experienced pathologist can very quickly scan a slide and find regions of interest for diagnostic purposes, but it takes quite a lot of training and lots of equipment. So there are solutions for digitizing this data, such as whole slide imaging, as well as ways to remotely operate microscopes. Nonetheless, these methods are significantly slower than having clinicians directly operate microscopes in person. And this actually also presents challenges for doctors to be able to keep up with caseloads, particularly in small hospitals where specialists might be in high demand, or for surgical cases where there's a lot of back and forth with the neurosurgical teams. Interestingly, reduced staffing measures during the COVID-19 pandemic actually might provide additional motivation maybe to operate microscopes and or review images remotely, which is a potentially interesting development. So there might be more of a push for this in the future. So I'm not going to be talking about my specific thesis work today, but something that occurred to me in looking at the state of neuropathology and informatics was that a lot of the challenges faced in bringing informatics into neuropathology are not unlike those faced by many researchers in basic science, even in molecular and cellular neuroscience. where we have large numbers of slides with many sections of tissue that also have to be analyzed in order to get to more quantitative data about whatever disease model we're studying. So just like in a pathology lab in a hospital, these slides might contain tissue sections or individual cells spread across the surface of the slide or a cell culture plate. And so here's just two examples from my own work where I'm also using histological methods to understand the disease processes at work in these individuals or groups of cells. So what I'd like to show you next is a series of what I think are really innovative responses to the challenge of obtaining informative microscopy data quickly, at scale, and with relatively low cost, all developed for use in the biological research community, the basic research community, but that I think has important implications for the clinic and in particular for neuropathology. So in 2016, a group led by Ann Carpenter at the Broad Institute published a technique called cell painting, which is a form of morphological profiling. And morphological profiling can be thought of similarly to drug screens, which you might be familiar with from pharmaceutical industry. So in screens, a large number of pharmaceutical agents are tested in many cells under certain conditions based on some previous data to test for a therapeutic effect. However, one chief strength over this of morphological profiling, which sometimes also referred to as image-based profiling, is that it's completely unbiased. And so that allows it to be broadly applicable to many experimental questions, and it's robust across variable cell types. And this is particularly important when studying the brain as a particularly heterogeneous organ compared to other tissues in the human body. So in this case, rather than working through as many slides as possible containing samples from different patients, as would happen in a pathology lab, researchers were looking for a way to identify biological patterns across many samples of the same experiment by extracting quantitative data from many microscopy images. And this is also an important innovation in advancing the use of microscopy research in basic science, because typically researchers will often just use microscopy data qualitatively in studies to show a phenotype. And so their solution enables a high throughput experimentation on adherent cell lines, which can be grown on multi-well plates, which is shown in the schematic. And these can be easily imaged in a similar fashion in large numbers of in a similar fashion to large numbers of microscopy slides on machines such as slide scanning microscopes, which are commonly used in the research setting to enable rapid imaging of many, many slides, which effectively digitizes your samples. So prior to imaging, an important feature of the cell painting technique is that they label cells with different fluorescent dyes, which are shown here, and they make visible various cellular components and organelles. And a large number of morphological properties and intracellular changes can then be assayed on the basis of labeling each of these structures at this single cell resolution. And so this microscopy data can then be sort of automatically acquired using a slide scanning microscope and then analyzed using open source pipelines that this group also developed that may incorporate machine learning to measure the morphological features of cells in this unbiased way. And so this has the potential to reveal phenotypes or biomarkers that might otherwise not have been able to be identifiable by eye, even by a highly specialized pathologist. So one tool in this image analysis toolbox is Cell Profiler, which I use a lot in my research to really good effect. And it's free software produced by the same group at the Broad Institute. And it has really broad applicability for processing large data sets of cytological images, which can be used to identify mechanistically relevant features of cells. So like within these nuclei that were automatically segmented by Cell Profiler, you can appreciate the nucleoli or clusters of RNA in the cells. When you look across all the different features that you can isolate from individual cells, you really quickly scale up to many different features. So you can look at the intensity of staining, you can look at the relative locations of different cells to one another in complex tissues, and you can quickly start to appreciate phenotypes that you might not be able to notice qualitatively. And so this has really incredible potential to be used not only as a research tool, but as a discovery tool for therapeutics. And towards this, the Broad Institute has recently established a new initiative called JumpCP for joint undertaking in morphological profiling with cell painting. It works based on this technique, and they have partners industry and as well as in nonprofits with the goal of generating a large cell imaging data set to enable discovery of drug pathways for precision medicine. But beyond this, I think the tools developed by this group have the potential for direct clinical use in applications like cytopathology, both within neuropathology as well as more broadly. And just a little bit more from this group, I think it's really illustrated well by actually very recently published collaboration between Ann Carpenter's group at the Broad and the Marti Lab, looking at stages of malaria infection based on actual patient samples of these blood smears, which you can see an image of here on the left. And so their approach actually enabled automated scoring of cells in the various stages of malaria infection, and they were able to improve upon the human efficiency and consistency of disease staging using previously annotated cytopathological data and a custom region-based convolutional neural network. Another cool thing that they did was to use deep learning in a flow cytometry experiment, which is doesn't have as much clinical relevance in pathology that I know of actually, but what they did was they used deep learning to identify just from bright field images that you can see of individual cells that were being flowed through this flow cytometer without any labels of any kind, no cell stage markers, no proliferation markers, to identify what stage of the cell cycle a given cell was in. And so this frees up a lot of channels for using other types of cell markers, which could be really powerful for identifying cell phenotypes that, again, you wouldn't be able to identify by eye because typically humans need some kind of cell cycle marker to do this kind of data analysis. And so finally, work from the Carpenter Lab has also been applied towards clinical trials for leukemia and lymphoma, identification for therapeutic targets to treat Ebola, as well as therapeutics to treat cerebral cavernous malformations, all based on this imaging data. And so another thing that I mentioned for clinical pathologists was the time constraint that is faced by clinicians, even after they might have gotten around the problem of digitizing their histopathological samples. And this is surrounding the use of hardware and software for navigating the digital images. So here I'd like to mention another actually brand new and very impressive open source image viewer for, which works with Python called Napari. And it's designed to be fast and interactive for use with viewing and annotating and analyzing large multidimensional data sets, such as histopathological samples. So on the left here, you can see this really big, it's 100,000 by 200,000 whole slide image, and you can zoom in and scroll around just as fast as an experienced neuropathologist would on a regular microscope. And you can also readily view annotations overlaid on these images. So it's a different histology image. And so if you want to do any manual or machine learning cell segmentation or label anything in your images, you can do this really easily within this Python interface and a relatively user-friendly GUI. And all of this also is open source, which is more often than not the case in clinical software. So I'm not saying this is ready for prime time for use in the clinic per se, but I think that cytotechnologists might soon be able to use software like this or future versions of it to not only increase their output and ability to screen more patient samples at low cost, but to identify potentially clinically relevant features of cells that could pave the way for finding new biomarkers of disease. So although deep learning has yet to play an active role in clinical neuropathology to date, I hope that I've been able to demonstrate some exciting applications which are ongoing in basic biological research that have significant potential to benefit the field and ultimately patients. And so as I conclude, I'd just like to highlight the Scientific Community Image Forum, which is a really great free resource for anybody working on bioimage analysis that people in this community might not yet be aware of, but it's for anybody working with any software or technique, especially in biology. And this was started by Ann Carpenter as well as Kevin Elisari under a newly formed NIH Center for Open Bioimage Analysis. And if you're on Twitter, you can check out the bioimage analysis hashtag, and you'll find there's really wonderful, supportive, and creative imaging and imaging analysis community that is very active. So thank you for your attention, and to Randy for inviting me to speak and talk about this cool stuff. I hope you all think it's as cool as I did. Thanks.